1998, one protein made cancer cells kill themselves. The lab was Institut Cochin, a low-slung concrete building on Rue du Faubourg Saint-Jacques in Paris. It was March, and inside, a small team led by virologist Mathieu Véron had been working in near silence for 3 years. They weren't looking for a cancer cure. Their focus was a chicken virus called TVV chicken anemia virus, a pathogen so obscure it barely registered outside poultry science. They were mapping how it hijacked cells, how it slipped past defenses like a thief in the night. But on that Tuesday morning, something shifted. A single microscope slide, placed under the lens by a postdoc named Claire, changed everything. Claire had been up all night, her eyes burning from the strain. She adjusted the focus, leaning in until her breath fogged the eyepiece. At first, she thought she'd made a mistake. The human cancer cell's breast carcinoma, aggressive and fast-growing, were behaving strangely. Their edges were bubbling, like wax melting under a flame. Their nuclei, usually dense and dark, were breaking apart, the DNA unraveling into fragments. She watched transfixed as the cells shrank, their membranes collapsing inward, until there was almost nothing left. And beside them, in the same dish, healthy cells sat, untouched, plump and unharmed. She called Mathieu over. He peered through the microscope, his fingers tightening on the edge of the bench. "Run it again," he said. His voice was steady, but his pulse was hammering. This wasn't supposed to happen. Not like this. Not so cleanly. They repeated the experiment. Same result. They tried different cancer lines: lung, brain, colon. Apoptosin, the protein they'd isolated from the virus, killed them all. No radiation burns, no chemo's brutal collateral damage. Just a viral protein, slipping into the cancer cells like a key into a lock, flipping a switch that said, "Die." By the end of the week, they moved to mice. The tumors were aggressive, the kind that doubled in size within days. 72 hours after injecting apoptosin, the tumors had shrunk by 90%. The mice were alive, scurrying around their cages, their fur sleek, their eyes bright. No weight loss, no lethargy, no side effects. The lab was silent. Someone exhaled sharply. Another whispered, "We just cured cancer in a Petri dish." But here's the thing about discoveries that rewrite the rules. They don't just change science. They threaten markets. In 1998, oncology was a $50 billion industry. Chemotherapy, radiation, surgical interventions, these weren't just treatments. They were profit centers, built on decades of research, regulatory approvals, and carefully constructed narratives about what cancer care had to look like. 3 weeks after the first successful mouse trial, the lab's funding was cut. The official letter arrived on a Thursday, slipped under Mathieu's office door. It cited budget reallocation. No explanation, no appeal. The team was given 48 hours to pack up their samples. Claire found her notebooks missing from her desk. When she asked about them, the lab manager just shook his head and said, "Protocol." Mathieu was called into the director's office. The man didn't look him in the eye. "Your position will be terminated at the end of the month," he said. "We appreciate your contributions." That was it. No discussion, no acknowledgement of what they'd found. Then, on a Friday afternoon, the lab director called an emergency meeting. The team gathered in the small conference room, the air thick with tension. The director stood at the front, holding a single sheet of paper. His hands were shaking. The rest of the notebooks, the raw data, the slides, everything had been shredded. Claire's stomach dropped. She thought of the mice, still alive in the animal facility. The director cleared his throat. "This research is too destabilizing to proceed," he read. "Further investigation is not in the public interest." The memo wasn't from the university. It was signed by a member of a pharmaceutical board, a man whose company manufactured some of the most profitable chemotherapy drugs on the market. The room was silent. Then someone laughed a sharp, disbelieving sound. Another person stood up, their chair scraping loudly against the floor. "You're kidding. You're kidding." The director wasn't finished. He slid the one surviving page across the table. It was the first draft of their paper, the one they'd submitted to Nature. At the bottom, in red pen, someone had written, "Retract," or "face consequences." They didn't retract. Instead, they buried the paper. Not in a drawer in a safe deposit box in a bank on Rue de la République. The team scattered. Claire took a job at a diagnostics lab, far from cancer research. Another postdoc left science entirely, becoming a high school biology teacher. Mathieu stayed at Institut Cochin for another year, teaching undergrads, publishing nothing. Then he quit. The last anyone heard, he was working at a veterinary clinic in Lyon, treating cats and dogs, his PhD gathering dust on a shelf. But the story doesn't end there. Because in 2003, a researcher in the Netherlands stumbled upon apoptyn again. His name was Lars, and he was digging through old university servers, looking for forgotten data on viral proteins. He found the original paper still unpublished, still buried tucked away in a folder labeled Archived 1998. He read it twice, then a third time. Then he replicated the experiments. This time, the world was ready to listen, or so he thought. When he submitted his findings to a top-tier journal, the paper was sent to the same pharmaceutical board member for peer review. The reviewer's response was swift, unreliable, not reproducible. The paper was rejected. Lars appealed. He provided raw data, video footage of the dying cancer cells, testimonials from other researchers who'd replicated the results. The journal stood by its decision, so Lars did something radical. He posted the data online, raw and unfiltered. Within weeks, labs in Germany, Japan, and the US began testing apoptyn. The results were the same. Cancer cells died. Healthy cells lived. The mechanism was elegant, precise, a molecular assassin that knew its target. So why isn't apoptyn in hospitals today? Because science isn't just about discovery. It's about money, power, and who gets to decide what's true. In 2005, a patent for apoptyn was filed not by the original team, but by a biotech firm with ties to the same pharmaceutical board member who'd shut down the research in Paris. The patent was granted, then it was buried. No clinical trials, no FDA approval, just a legal wall built brick by brick to ensure no one else could develop it. And that one surviving page, the first draft of the original paper, it's still online. You can find it if you search apoptyn 1998 Paris. It's just a single page, yellowed at the edges, with a title that reads, Selective Induction of Apoptosis in Human Tumor Cells by a Viral Protein. No figures, no data, no graphs, just a title, an abstract, and promise of what could have been. Here's the question no one asks. How many other discoveries have been erased before they could change the world? How many labs have been shut down? How many scientists discredited? How many papers buried not because the science was wrong, but because the truth was inconvenient? Institut Cochin is still there, on Rue du Faubourg Saint-Jacques. The building looks the same. The microscopes are newer. The funding comes and goes. But if you walk past at night, you might notice something strange. The lights in the virology wing stay on late. And sometimes, if you press your ear to the glass, you can hear the hum of a centrifuge running long after everyone has gone home. It's a quiet sound, persistent, like a heartbeat. Search apoptyn 1998 Paris. The one surviving page is still online.